As I mentioned, we're publicly traded. Our ticker symbol is CAPR, and we're listed on the NASDAQ. So Capricor has been in business for about 15 years. We're originally out of Johns Hopkins University. Our founder is a gentleman by the name of Dr. Eduardo Marban. We relocated to Los Angeles about a decade ago now, where we're housed at Cedars-Sinai Medical Center. And the company has 2 programs in development. The first is our lead asset, which is a cardiac cell therapy, which I'm going to talk about, which is now in clinical development for the treatment of Duchenne muscular dystrophy, as well as an exosome therapeutic that we have multiple indications that we're working through preclinically. I won't belabor this slide. It's kind of a quick snapshot of where Capricor has come from. We have a very strong intellectual property portfolio out of Johns Hopkins, out of the University of Rome, and out of Cedars-Sinai Medical Center. The majority of my talk today, I'm going to spend time updating you on our cell therapy program for the treatment of Duchenne muscular dystrophy. We recently read out phase 2 results on an interim analysis basis, which were very exciting, and I'll talk a little bit about that. And from a financial perspective, we've raised about $50 million to date in equity financings and another $45 million in non-dilutive funding that has funded the company to date. And those sources of capital are primarily from the National Institutes of Health, from the Department of Defense, and from the California Institute for Regenerative Medicine. We also have a very interesting partnership that I won't talk about today, but just to tell you about briefly, we have an interesting partnership with the United States Army Surgical Research Center, where they're working on testing our exosomes in different varieties of trauma injury. So our lead asset, known as CAP1002, it's an allogeneic cardiosphere-derived cell. It's a progenitor cell that's unique to the heart, and it's been in about 150 patients to date. Capricor, in our early days, spent time in heart failure, post-myocardial infarction, and large cardiovascular indications. We now have shifted over the last 5 years into Duchenne muscular dystrophy, where we're now in a phase 2 study. About 2 years ago, we read out positive proof of concept study data in that phase 1 study. And in the exosomes, we're exploring a variety of different inflammatory and fibrotic indications. So a little bit more about CAP1002. It's not a stem cell. As I mentioned, it's a progenitor cell. It's derived from human heart tissue. We obtain hearts from organ procurement organizations, and essentially the MOA functions by secreting exosomes. Exosomes are packed full of microRNAs, yRNAs, certain non-coding RNAs, as well as proteins, which drive the biological effect of our cells. But one of the takeaways that I want you to remember is that we believe our cells to be functioned by immunomodulation. That means regulating that bad inflammation and allowing that good healing inflammation to occur. And we've seen this now clinically and, of course, preclinically over the last decade that we've been running clinical studies. So Duchenne muscular dystrophy. DMD is a completely fatal disease. I'm sure everyone's heard about it. It's kind of become the darling of the rare disease orphan space, but it's a very devastating disease that afflicts about 15 to 20,000 boys in the US. Essentially, they don't code for a vital protein called dystrophin. This protein acts as kind of the shock absorber of the cell. I always like to draw the analogy to the bumper on your car. But when you don't produce dystrophin, essentially you have a muscle wasting disorder that continues to occur, eventually necrosis, and the body and the cells aren't functioning the way they should. And when there's many companies treating Duchenne muscular dystrophy in the clinical development stages, and a lot of them are addressing the genetic component. Capricor is addressing a little bit more of the downstream complications of the disease around skeletal, cardiac, and respiratory improvements. But there's a variety of different things going on in this space. So a little bit more about the progression of the disease. It's usually diagnosed at around 2 or 3 years of age when boys begin to lose their milestones. Essentially, they start to lose their ability to climb upstairs, eventually get up off the floor, to rise. And you can see the arrow here drawn to where Capricor's target population is a little bit in the later stage of the disease. I always define them as the non-ambulant patients, but essentially these guys are no longer typically walking at this stage. They're on high doses of steroids for the most part, which carry with them a lot of different complications. And there's really no therapies that have been shown to address these complications to date. Only 2 approvals in all of DMD. Number 1, Sarepta's drug, Exondys 51. It only treats 13% of boys with DMD. And number 2, steroids. Steroids have been shown to extend the quality of life with boys with DMD, but they do carry with them a lot of long-term side effects, especially when you're on them many years. So a snapshot of Capricor's regulatory developments to date. We have orphan drug designation, of course, with Duchenne muscular dystrophy. We also qualify for a rare pediatric disease voucher, where upon approval we would receive the voucher, which I think everybody knows these sell for very high dollar amounts. They're very interesting programs. So upon approval we would obtain that voucher, but we do qualify for it. And lastly, everybody in the room knows what RMAT designation is here at the Cell and Gene meeting, but we do have RMAT designation for this as well, which allows us to go discuss this program with the FDA throughout its development, and we've done just that over the last few years. And we actually have an upcoming meeting with them in Q4, which we'll be providing updates on later this quarter. So obviously a very exciting program and a very, very important regulatory designations that we've obtained. So as I mentioned, Capricor completed a phase 1 study in DMD about 2 years ago, and we designed a phase 2 study now. And I'm going to talk very briefly about some of the data that we saw from this phase 2 study. It was originally designed as an 84-patient phase 2 study. We were positioning it to potentially serve as the registration study. And what we did after about 25% of the patients became enrolled, we decided to do an interim look at the data. But this really walks you through essentially what we're looking to do. We were treating our patients every 3 months with 150 million cells administered IV. So basically, the cells would be administered quarterly in a repeat dosing fashion so that a patient would receive 4 doses over the course of a year. It was placebo-controlled, so they would either receive blinded placebo or drug product. And we would measure them out at different time points, basically 3, 6, 9, and 12, looking at a variety of different endpoints, focused mainly on a skeletal muscle endpoint known as the performance of the upper limb. I think I have a slide on it, but I'll talk about it in a few moments. It was conducted at 9 centers throughout the United States. A lot of the big Duchenne centers obviously ran this study, and our national PI is a gentleman by the name of Dr. Craig McDonald out of the UC Davis. He is now presenting updated data on this trial tomorrow at the World Muscle Society meeting in Copenhagen, and Capricor will be hosting a conference call on Monday morning at 5:30 in the morning Pacific if you want to wake up early, or if you're on the East Coast at 8:30. So I have one slide, and this is the main primary endpoint that we looked for in this study, and this is known as the performance of the upper limb. And a little bit more detail on the performance of the upper limb measurement. Not many people probably know about it. It was designed by clinicians, patients, doctors, and families in the Duchenne community to measure daily tasks and of living for patients with DMD, essentially looking at the shoulder, the elbow, and the hand, and their ability to continue to complete milestones around this. So it could be as simple as moving a can across a table, which might simulate drawing a pencil or writing, moving your joystick on your wheelchair, drinking, all those daily tasks which we take for granted. Of course, these boys with this devastating disease slowly lose their ability to do so. So this test was designed specifically for DMD. It's an independent measure. And we looked here. Now you see data at 3 and 6 months. And this is on approximately 14 patients in our study, randomized, double-blind, placebo. Placebo in blue is doing what you would expect, which is continuing to decline. And when you look at the natural history of the disease, you do see a continual decline in their PUL scores in the data that's been published so far to date. And again, you see somewhat of a good response, again in small numbers, with the patients treated with CAP1002 at 6 months with a p-value of 0.0389. So we were very excited by this data. We've now taken it to FDA. We're now meeting with them on an end-of-phase meeting for this program. And this is also the second trial now where we have seen these changes in the performance of the upper limb. So obviously very exciting, and stay tuned. I have more data which is available on our website where we saw improvements in respiratory function, trends towards improvements. And again, the lungs of the patients with DMD, for the most part, are functioning fairly well, but it's the diaphragmatic muscles that tend to lose their strength over time. And then we also saw improvements in cardiac changes, which we also have found very interesting. So again, a very robust data set that we're positioning to take to FDA. We're exploring the potential for accelerated approval. That's just what RMAT offers companies. At least that's why I believe they put it in place. So that is one of the possibilities. We're excited to hopefully bring this program to FDA and move it forward as quickly as we can to patients. This is one of the last data sets I have. This is an independent measure outside of the pull looking at additional skeletal muscle changes. One's called grip strength, just like it sounds, and tip-to-tip pinch strength. This is measuring hand and mobility function. And again, you can see trends towards improvements, which supports our performance of the upper limb data. So in summary, as I mentioned, we had changes in skeletal muscle, PUL 2.0, that's what it's known as, the PUL 2.0 assessment. Grip strength improvements, which FDA specifically asked for in our meeting. They wanted us to come back with independent supportive measures around the PUL. Respiratory trends towards improvement in expiratory and inspiratory flow, as well as anterior wall thickening changes, and LV myocardium mass, measures of cardiac function. So we're excited to continue to discuss this program, so stay tuned for updates from Capricor this quarter. I believe this is one of my last slides. So as I mentioned, we have 2 products in the pipeline. Capricor's second is an exosome product. Exosomes, I think everybody knows what they are, but they're basically emitted from all cell types. They're a nano-size lipid bilayer vesicle. Exosomes are packed full of microRNAs, yRNAs, and we at Capricor right now derive our exosomes from our cardiosphere-derived cells, but many companies are looking at a variety of different exosomes. Ours are very unique in the fact that they've come from the heart. We're starting to unpack exactly what's inside of them, how to characterize them, how to make them in larger scales. It's a very exciting field right now that's starting to develop. Not many companies are in the clinic, but a lot of companies are starting to explode, especially with respect to publications, grants, and all that. So I'm happy to talk with anybody after this about where Capricor's at in our exosome development. And as I mentioned, we're housed on the campus of Cedars-Sinai Medical Center, a really great facility. Cedars owns a portion of Capricor with respect to equity investments over the years. Of course, they're our licensor. We have a great relationship with them. We have a phase 1, phase 2 manufacturing facility located in the building, along with our research labs, and we've been housed there over the last decade or so. So a great relationship with Cedars, and obviously in the Los Angeles biotech community, which has become very exciting. And I think that is my last slide. I want to thank ARM again for hosting, and if I can take any questions, I think I have a minute or two, happy to do so, or talk outside.